Thank you very much to OIS for inviting me to present our story. We are a publicly traded company, so I will be making forward-looking statements. This is our pipeline, and as you know, we have a lead program in Retina called GB102, which is a 6-month formulation of a tyrosine kinase inhibitor that we tested in wet AMD. But we also have 2 new programs. One is in retinitis pigmentosa, and one is in geographic atrophy. And I'm going to spend more time today detailing these programs. We also have a 6-month sustained release formulation in glaucoma, which is a new chemical entity, which will be injected in the vitreous, as well as a brand new gene therapy for a very rare disease called MPS I. And that disease presents as a corneal clouding, which we intend to correct by injecting a gene therapy coding for a missing protein called IDUA in these MPS I patients. So for our sustained delivery technologies, we work with small molecules, and we have basically 2 platforms. One is microparticles that we can make, aggregate, or not. And as well as an implant capability for which we developed not only the manufacturing capabilities as well as the implant injector in-house. So the first program for today is a GB601, which is a cGMP analog for the treatment of retinitis pigmentosa. So the role of cGMP is very well known in a number of disorders, and the accumulation of cGMP triggers the death of photoreceptors. And you can see here in multiple animal models in IRDs, you have this white accumulation, the white stains, is accumulation of cGMP in the ONL, which is not seen in the control animal, as you can see here. So what we are intending to do here is to basically use GB601 to block the negative impact of this accumulation of cGMP and protect the photoreceptors against cell death. So there is an amount of evidence, preclinical evidence, showing that it does work. So in this RD10 mouse model in retinitis pigmentosa, you can see that the not treated animal has almost complete destruction of photoreceptors in the ONL, as opposed to 2 compounds that we tested from our library, which result in a protection of these photoreceptors, as counted by the number of photoreceptors rows, as you can see on the right-hand side. So anatomically, it seems to work, blocking this cGMP accumulation, but it also works functionally. And here you can see the ERG data in these animal models, where the not treated animals clearly behave very differently to the animals that were treated with our lead compound, which we intend to develop in a sustained release formulation. So what we are intending to do here is quite interesting, because it is actually mutation agnostic as a new approach, as opposed to genetic approaches which require a particular genotyping of the patients and result in quite small patient population. Here, this approach, we mapped the different mutations in RP, could be applicable for around 1/3 of RP patients. 50% of LCA patients and 85% of Stargardt patients could potentially benefit from this unique therapy. We'll develop it on the long-acting formulation, and we are targeting a 3 to 6 month duration to start with. The second program that is new is this GB701, which is a factor B inhibitor. It's a small molecule, and we are initially intending to develop it for geographic atrophy. So there is a lot of evidence about complement inhibition. The session this morning told you everything you need to know about complement inhibition. And the excellent work done by Apellis and Iveric Bio are just validating that this is a very nice target for a therapeutic intervention. But we believe to get the scale of use in clinical practice, longer duration will be required than monthly or bi-monthly injection for this vast amount of patients. So what we are doing is developing this small molecule factor B in collaboration with In Silico Medicines, which is an AI-powered drug discovery company, which generated a whole library of potent molecules for us. So we decided to target factor B for a number of reasons. One of them, using small molecules, is going to diffuse very well in the target tissue. The second reason was that factor B is upstream to C5 and C3, and therefore by blocking it, we're going to block the whole cascade. And the other reason is factor B is not expressed in very big quantity in the eye, and therefore it's very amenable to a sustained release, low dose approach, as opposed to C3 or C5, which are expressed in a much more amount in the eye. And finally, we want to also develop a sustained release formulation to make sure these patients don't have to be injected all the time, and we are aiming at a 6-month formulation. So the first indications for GB601, sorry, as I discussed before, it will be retinitis pigmentosa, and then we will expand into LCA and Stargardt. For GB701, we want first to look at it in geographic atrophy, but obviously after going to intermediate AMD. Thank you.